Okay guys, this is where the fun really begins in specific immunity. It's our third installment of the lecture, and now what we have are differentiated T cells and B cells. They have their own jobs to do. They've been activated and they know what antigen they're after. So now we're going to talk about what happens. They're activated, what do they do? How do they work in the immune system? These are the different types of T cells and B cells that we're talking about. Again, we're going to talk about helper T cells, regulatory cytotoxic T cells. What do they do? We're going to talk about antibodies and what they do. This T cell response, is another word for it is cell-mediated immunity. That's because it involves several subsets of T cells who their job is dictated by the antigen presenting cells that activated them to start with. So what I mean is those are the different types of T cells we talked about, cytotoxic versus helper. What do they do? What actions do they have? And it's what they do that mediates the immunity. So that's why it's cell mediated, because these are specific cells that are doing the job. All of them produce cytokines. Those are chemical mediators we talked about with innate immunity. And they have a large spectrum of biological effects. So let's talk again about helper T cells. We talked about them a little bit before. They play a central role in regulating the immune reactions. That's because they have those CD4 markers. CD4 markers respond to antigen on MHC2 receptors. When they have antigen presenting to them, they can then activate B cells and other T cells to do their job. They're also involved in activating macrophages. So again, helpers, they help to activate other cells that then do the job of the immune system. So they are only indirectly involved in the immune system because they don't actually do any of the killing. The regulatory T cells, again, they act to control the inflammatory response. So they are involved in preventing autoimmunity, so preventing your immune system from attacking itself. They make sure that the immune response does not respond inappropriately to your normal microbes too, which is also very important. Remember, we have these normal microbes that colonize the body and help to prevent infection, so we don't want the immune system to attack them. That's the job of regulatory T cells. The cytotoxic T cells, these are the ones that are directly involved in killing microbes. They are cytotoxic, so that means that they are able to kill specific microbes that they are targeted to kill. They target cells targeted by T cells, cytotoxic T cells, are usually cells that are infected by viruses. Cancer cells can even be targeted by the cytotoxic T cells, and cells that are non-self, so from other animals and humans. How do they kill? Well, they kill by releasing different chemicals. Some chemicals that they release are called perforins. These, just kind of based on the name, you can tell what they do is they punch holes in the cells that they're targeting. So it's like perforins perforate. So they're proteins that bind to the surface of the microbe, punch holes in them, and then that causes ions in other parts of the inside of the cell to start leaking out. And what it also does is it creates a passageway for these other chemicals called granzymes to get into the cell. What granzymes do is they attack proteins of the target cells. So once they're able to enter the target cells, they find certain proteins, attack them, denature them, and then the cell is targeted to die. Okay, so after attack with these two chemicals, these targeted cells then go through death by apoptosis, which is programmed cell death. Okay, and then this is what it would look like here. These are cytotoxic T cells attaching to the outside of their target cell, and they're starting to release those two different chemicals to kill it. Now another type of lymphocyte that's related to T cells that has, as we call it, orders to kill, are natural killer cells, or NK cells. Okay, again, natural killer cells are related to T cells. They're lymphocytes just like them, but they lack specificity for antigens. So we don't really consider them part of the specific immunity or cell-mediated immunity because they don't possess antigen receptors. They don't have a way to target them to any specific microbe. But they are first killer cells, the first killer cells to attack cancer cells and viruses, cells infected by viruses, in a manner that's similar to cytotoxic T cells. So even though they're not specific, they don't have specific antigen receptors, they're an important part of the immune system because they also attack cells that are infected. This table right here is showing you differences between B cells and T cells. We already talked a little bit about the T cells, so the next thing we're going to do is talk about B cells and what they do. And the main thing that they do is make antibodies. So the important thing to get out of this next part is, what do antibodies do? So here we are, the B cell response. Well, again, B cells, their main job, once B cells are differentiated into plasma cells, those plasma cells produce antibodies. Well, how do antibodies do anything in the immune response? Well, antibodies, again, are made specifically for specific antigens. They're made to bind to those specific antigens. When they do bind to a specific antigen, that what that does is that signals even more antibodies to come in, bind to that specific antigen. And then these are the four things that they can do in order to kill that microbe or work in the immune system. So we're going to talk about opsonization, agglutination, neutralization, and antitoxin. Alright, so this is opsonization here. I mentioned when a microbe enters the body that have antibodies specific for it, those antibodies bind to the microbe, so they bind to the antigen, and they line the whole surface of that microbe. And once they line the whole surface of that microbe, that makes them more likely to be taken up by phagocytosis. So that's what we call opsonization. The bacteria have antibodies surrounding them, and that makes them taken up by phagocytosis more readily. Neutralization generally happens when we're talking about viruses. In neutralization, antibodies again bind to the surface, but this time they're binding to the surface of viruses. Okay, binding to the surface of the viruses then blocks those receptors, those spikes on the surface of the virus, from being able to attach to host cells, and it prevents them from getting inside of the host cells and infecting them. So it effectively neutralizes the viruses. It prevents them from being able to get in and cause an infection. We also mentioned agglutination. Okay, agglutination happens when, again, the antibodies are attracted to the antigens on the bacterial cells, but this time it actually links bacterial cells together. So the antibodies don't just surround the outside of the microbe, they can clump or cluster those bacterial cells together. Now, these clusters then that are created, these clumps of bacterial cells that are created, enhances phagocytosis because now all of these microbes are all in the same location. And when a phagocyte, like a macrophage, comes along, it can eat a whole clump of bacteria instead of going around finding individual cells on their own. The final thing that we're talking about here is antitoxin. So instead of attacking a whole cell or a whole virus, antibodies can actually recognize toxins. We mentioned that before, that bacterial toxins are a great antigen because they are unique to the bacteria and they can elicit an immune response. So because antitoxins can be an antigen, that means there are antibodies that can be produced to it. So in this case here, the toxin that's produced by the bacteria is recognized by the antibodies, and then the antibodies bind to it, neutralizing the effect of the toxin. And this is great for diseases that are caused by bacteria that produce toxins. For example, botulism. Botulism has its effects by producing a toxin. So people that have been exposed to it before, they're producing antibodies that bind to the toxin and neutralize it, so you can use those antibodies to treat people that become infected with botulism. There are many different types of immune globulins or antibodies out there, but there are just two that I want you guys to focus on because these are the most important for what we're talking about with the specific immune system. First off is IgM. Again, Ig stands for immune globulin. M is the version. IgM are the first antibodies produced in response to an antigen. So this is involved in what we call the primary immune response, the primary antibody response. Okay, once B cells have been activated by a certain antigen, the antibody that they produce is generally IgM. These are the ones that you find on the surface of B cells. IgG are involved in long-term immunity. Okay, that means that these are memory antibodies. So they stick around for a long time, are present in your blood, and can, when this microbe re-enters your body, can be activated again and will bind to it. So we call that the secondary response. And this is the IgM here. Alright, this can serve as the B cell receptor, but this is the one that's produced in response to that first presentation of antigen. This over here, this is IgG. This is the one we're talking about for long term. Okay, these guys are the ones that are produced in high quantities that are specific to that antigen, and they're the ones that are involved in the reactions that we talked about before: opsonization, neutralization, antitoxin. Okay. Now, one thing that's important for us to understand is, because this specific immune system is such an important part of health, one thing that we can do in the medical field is to see how much of a response a person really has to that microbe. We can do that by seeing how much antibody a person has produced to a specific microbe. So how much antibody is present at a certain time is called the titer. That's the concentration of antibodies in the blood serum. The higher the number of antibodies that you have, the more of a vigorous response, immune response, you will have to being exposed to that microbe. If you do not have many antibodies at all, if it's in low levels, okay, then the response to that microbe is going to be slower. So it's a measurement over time to see how much antibody you have and to see the reaction of your immune system to an antigen. I mentioned before the different types of antibodies and primary versus secondary response. In the primary immune response, this is when you're first exposed to an antigen. So this is when B cells first become activated. It's the first antibodies that they produce. So the primary response is that first time that you're ever exposed to an illness. So let's have an example here, an example of chickenpox. you're a kid, you get chickenpox for the first time, and what we're going to find out is that it would be the only time. So when you have chickenpox, your immune system starts to produce antibodies that are specific to the chickenpox virus. Okay, you build these antibodies, it gets chickenpox under control, and then you go through something called a latent period. Okay, the latent period is marked by a lack of antibody production. This is where antigen is concentrated in lymphoid tissues. As your plasma cells start to produce antibodies, that titer increases to a plateau and then tapers off to a low level. So again, what's happening is you're exposed to the antigen, your B cells get activated, and they have to work to produce the antibodies. So it takes a while for the antibodies to really get produced, but then finally the plasma cells get in a rhythm, start to produce a lot of antibody. As the antibody levels build up, that reduces the infection, reduces the amount of virus. So the less virus you have, the less you need antibodies. So production of antibodies slows down because you don't need as many of them. After a while, again, you're not producing very much antibody to chickenpox. But if several years later, if you're exposed to chickenpox again, those few antibodies that were still there will respond to the presence of chickenpox. So this is the secondary or amnesiac response. This occurs when the immune system is exposed again to that same immunogen, that same virus, same bacteria. This can be months, can be years, could be decades later. At that point, the rate of antibody synthesis skyrockets. Okay, it's greatly increased over the primary response. And that's because you already have the ability, you already have antibodies there. B cells know how to make the antibodies. So as soon as you're exposed to that same microbe again, production goes into a high rate because your immune system is primed and ready to go. If we put this into a graph, you can kind of see what's happening with the primary and secondary response. Primary response, again, right at the beginning, you're first exposed to the antigen. So in this period, latent period, your body is trying to produce the antibody. It's picking out the right antibody that binds to the antigen. Your plasma cells start to bind, start to produce the antibodies, and eventually come to a peak. And once those antibodies are produced in a high enough quantity, that gets the infection under control. Okay, and when the infection's under control, antibody production goes down. Now this period right here, this is between exposures. So this can last months to years. Once you have a second exposure to that antigen, to that microbe, antibody production gears up in a high gear, and we have a lot of antibody produced. Again, until you get the infection under control, and then it can ease off again. Thank you guys for being patient and watching the specific immunity lectures. Don't forget, if you have any questions, to post your questions on the discussion board for specific immunity.